Next is Katherine Perkins.
Hi, everybody. I'm Katherine Perkins. Bob Wong picked the title of my talk, The GABA Response Enigma. So I decided to stick with that topic. I came to Downstate in 1990 with Bob, and I've been here ever since, and it's my home. So most of us, when we think of GABA, we think, oh, GABA's the major inhibitory neurotransmitter in the brain. And we think, I know GABA, that's the one. Chloride goes through the channel, and it hyperpolarizes the cell, and that causes inhibition. But it turns out that sometimes GABA causes a depolarization, and it can even be excitatory. So I'm going to talk about that today. So you can see here on this slide, let me see if I can make this little pointer work. Yes. Okay, so this is an electrophysiology recording from a CA3 pyramidal cell. And I'll move my little pointer over here. So this is a hippocampal slice taken from a guinea pig, but this phenomenon I'm talking about also happens in rats and mice. So right here, this gray line is the CA3 pyramidal cell layer, and that's what this recording is taken from. And on this particular slide, this is a sharp electrode recording, so you can see the electrode here just stabbing the cell. So when we put 4-aminopyridine into the bath solution, we get spontaneous GABA responses, and we call these the giant GABAs, or GPSPs. And in this particular slide, the glutamate transmission is blocked, yet you still have these spontaneous, happening every 15 seconds to 30 seconds or so, these spontaneous GABAergic events. And blown up in B there, you can see that each of these responses has three components. GABA A response is the early one, a depolarizing GABA there in the middle, and a GABA B response is the late response. The GABA B is a potassium channel. So it looks like here that the depolarizing GABA happens before the GABA B, but whole cell recordings in my lab show that those peaks actually coincide. The depolarizing GABA and GABA B overlap exactly. So if we put on a GABA B antagonist, it's going to block that third component. So what's going to happen? Is depolarizing GABA actually going to be excitatory then? So in this next slide now, glutamate transmission is intact. So you can see in A that here would be the depolarizing GABA, and it's actually triggering these bursts here. And we know, even though we're just recording from one CA3 pyramidal cell there in this top line, that second line is a field potential recording. And because we have a field potential, we know that the population of CA3 pyramidal cells are all firing together, that this is an epileptiform event. So then to that question, what happens if we block that GABA B response? So if you look down here in B, this CGP 55845 is a GABA B antagonist. And when we add it, you can see we get lots more bursts. And you can see there in the field recording that this is a population event. So over here in C, you can see as we add the GABA B antagonist that the number of bursts goes up in every slice. And what do we think is happening? So we think there's this group of interneurons here that are exciting one another to produce a population event so that GABA is landing on all these pyramidal cells at the same time. And then the connections between the pyramidal cells are actually responsible for these bursts. But it turns out that the length of this discharge is actually the length of the depolarizing GABA. So when we add the GABA B antagonist, the depolarizing GABA we saw actually gets longer. So that's an interesting feature that's going to come up a little later too. On my slide here on the right-hand side, you can see that that's a schematic of the GABA A channel. So it's got these 5 subunits. And before I was saying, we think of the GABA channel as a chloride channel, but we've known for a long time that both chloride and bicarbonate go through the GABA channel. So the one that's been studied, it's about a 5-to-1 chloride to bicarbonate going through that channel. So in these experiments, I'm actually using whole cell recording, so I can change the ionic concentration inside the pipette, and then the solution I put in the pipette is actually going to get into the cell. So what I wanted to know was, what ions are carrying this depolarizing GABA. So now we've got GABA B antagonists in there and glutamate antagonists in there. So we've got a biphasic response. So this is outward current, inward current. So this is what I think of as the GABA A followed by the depolarizing GABA there. And then you can see I have the pipette solution here, so I manipulated how much chloride and bicarbonate is going to be in the pipette. So, this is, let me see, get my little animation going. So here we have the outward current, chloride, that's going to be chloride going into the cell. And it's followed by the inward current. And I wrote here bicarb coming out of the cell. But both the early component and later component are going to have chloride and bicarbonate both moving. But I'm showing you here that this second component is much more sensitive to how much bicarb I put in the pipette. And then you can see in B that if I load the cell with chloride, now I've got an inward current here followed by an outward current. So I've been able to completely reverse the polarity by manipulating the anions in the electrodes. So here we've got the inward current first, chloride's coming out of the cell, and the outward current, bicarb, going into the cell. So one of the hypotheses for the depolarizing GABA was just, it's chloride accumulation, and then we can see that bicarb current. But by showing that I can completely reverse it by manipulating the anions, that makes that hypothesis much more unlikely. I had a paper where I went through all these various ways of showing that chloride accumulation does not account for the depolarizing GABA. So my question is there a subunit configuration that would give us a channel more permeable to bicarbonate? So you can see that there are 5 different subunits. Each one has many different isoforms. And knowing that, for example, alpha has 1 through 6, would there be a conformation there that would give us a channel with this higher bicarb permeability? And then would that be something we could target with drugs now if we knew what that conformation was and just block the depolarizing GABA as an anti-seizure medication? Right now, our GABA antagonists block both the GABA A and the depolarizing GABA.
30 seconds.
Thanks. Okay, so I was thinking, it would be nice to have a cell. So I showed you in the CA3 pyramidal cell that you have both the hyperpolarizing and depolarizing components in the same cell. But if we wanted to, say, do single-cell RNA sequencing, wouldn't it be nice to have a cell that had only the hyperpolarizing or only the depolarizing GABA? So I've been looking at interneurons, and I've been doing these cell-attached recordings, which is pictured here, so you don't actually rupture the membrane now, and you don't mess up what's actually happening in the cell. So these are 2 different interneurons. You can see in the top panel that when the 4AP event happens here, this interneuron's excited, whereas this one's inhibited. And it turns out, I did a methods paper for cell-attached recordings. It turns out you can record membrane potential cell-attached. It's kind of like you're using the cell's own channels to pass current there without rupturing the patch. So this top row is an interneuron from stratum lacunosum moleculare, and I've found 2 types of those interneurons. And you can see that one of them has this pure depolarizing response. So it would be neat if we could do single-cell RNA sequencing there. And the other one has the mixed response similar to the CA3 pyramidal cell. These are little raster plots of what the firing looks like if we're recording actual potential currents like in the upper panels. I didn't show here what this second interneuron would look like, but what happens in those is you get a pure hyperpolarizing response out of these interneurons that are inhibited during the GPSP. And I will stop there. Thanks.